All right. I think we're ready to kick it off. Steve Hoffman.
Excellent. So Beth, thank you for this opportunity, and many names and friends that I see from the past are here. We're here to talk today, a webinar to go through one of the emerging programs in our biomarker consortium program here at FNIH. And Beth has asked me to do some introductory comments to just level set. Many folks that may know us or know myself, I'm Steve Hoffman. I'm one of our vice presidents in our science partnership efforts, oversee our neuro, our I&I, and our rare disease efforts, and currently acting director of the Biomarker Consortium. Been here for 12 years, so I've certainly crisscrossed many paths with many of you in the programs and projects that we've done here at FNIH. Really appreciate everyone's time and efforts and attending today to hear about this exciting opportunity and partnership. So you can jump, Beth, to the next slide. Just wanted to make sure I level set about the FNIH. For those that may not know us or not interacted with us, we're our own 501c3 nonprofit organization. Really were enacted by Congress in 1996 to support the mission of the NIH, both through financial means, but also as a coordinator and convener of public-private partnerships to do more to enhance the NIH missions, to move forward, in the case here with the Biomarker Consortium, on biomarkers
Yes,
and
got through
tools
here.
and
Yes.
endpoints.
Yeah. Got the gel.
Hold
Let me
on
see.
one minute. I'll see who we can mute for a second. Ask those that are not on.
Press.
You got control, Beth. So, we certainly have taglines here and FNIH's mission to build bridges through breakthroughs. The whole idea that partnership is really the way forward in a lot of these efforts to accelerate new treatments, diagnostics, impact on patients' lives. That one organization alone really can't do that. The shared risk, the shared resources is really what we're all about as a neutral convener and facilitating the expertise, the brain power, the financial resources, the resources and samples and data, and bringing that together in a pre-competitive, open, broad sharing way is really what we are about at FNIH, and the Biomarker Consortium is one of those programs. Jump to the next slide, please. How do we do this? And, we feel we have a proven track record of success. By far and away, we're in these early parts here where we have a concept within one of our programs. We're listening and convening stakeholders, just like we do today. Ultimately want to get to that impact on patients' lives, but it takes all of you and want to hear from you and feedback and understanding how we design these projects in a way that can be most effective, that can return the investment across the different sectors that we're here, the patient advocacy, the industry sector, the basic research sector. Good partnerships really have a piece of all of that. We play that role of all the parts and pieces, activating that partnership, stewarding finances, contracting, fundraising, programmatic project management. We do a little bit of all of these, and then really are also part of the communication piece to get those results out there broadly. Again, pre-competitively, open data sharing, broad data sharing, and as quickly as a means as possible to move the field, and particularly back to the ultimate patient impact and how do we change lives and quality of lives. So these impact of these projects, as much as nuts and bolts as they may be, really has a multiple and diverse impact across the field. Next slide. And so you may know us. You may know us on many different types of programs. The Accelerating Medicines Partnership, a little bit more discovery-based programs. We were responsible in some aspects of managing the ACTIV program when we were in the pandemic on all aspects of the vaccine, the therapeutics, the variants that were there. We're doing some outstanding work in rare diseases now, the Bespoke Gene Therapy Consortium under our AMP program. We have large cancer programs and lung map and platform trials and the PAC biomarker effort that you see in cancer therapeutics. And also one of our big bread and butter programs has been a long-standing, about 20 years now, the Biomarker Consortium. And the Biomarker Consortium is really about validating biomarkers, really about moving bring tools, endpoints, adoption into clinical trials to accelerate, go from diagnostics to prognostics to monitoring to treatment response. These types of biomarkers are critical to accelerate drug development, critical for clinical decision making, and that's really the focus where this project is today in the biomarker consortium and moving this effort forward. So next slide. So, what should these projects always be? How can we be successful? We want to generate actionable information, again, that can lead to adoption and use in trials. We want to build the trust. We want this data set to be broadly shared with everyone and really build trust amongst the community, amongst the partners, that this is truly validated, strong data that can really lead to adoption and use of the different biomarkers, the different data sets that were there. Talked about democratizing that data access broadly. A lot of these come up with guidances, clinical guidances, regulatory guidances come out of a lot of these projects. And I think that's where also we want to do that here. And not only we build that leadership, not only within our own organization, but obviously bringing together the partnerships, the project teams, the steering committees, the sub teams, and having that joint expertise, both business-wise and scientifically, to provide that leadership is really what the BC is about and what this project is. What I'm excited about for this program in particular is it mirrors a previous biomarker consortium project we did in RA, where we leveraged an NIH clinical trial in support. And we think we can do that again here. We build on top of some biomarker work using those existing resources, those existing financial support by our big partner. We can't get away from the NIH letters on the back of our name. We want to be aligned and strategize with the NIH and all those efforts. That's really part of our main mission and vision. And I think this project, you'll hear more about how this aligns and what we want to do that really touches base with those roots, but also can be operationalized across different sectors. So that's kind of sort of my intro. I didn't have very much more there as a level set. What I'm really excited about, and what is an important component of every project we do, is patient with lived experience voice, and making sure that our projects are designed and incorporate what helps our patients, where they think we should be going. They're just as valuable of a partner at every stage of design, development, implementation, and outcome of every one of the projects. And we are super pleased, and you can jump to the next slide, to have Steve Taylor with us. We know Steve's now, overseeing the Arthritis Foundation. He's been a long-standing partner in many of the programs with the FNIH. I've known him for years. Ages with the Sjogren's Foundation, really put Sjogren's Foundation now with Kathy and Janet on the map, but Steve's foundational work there is just outstanding. But this is simply an advocate and a guy that's been involved in the nonprofit world, involved in many diseases for years. But today he provides us a critical patient voice in gout. And so his voice resonates, his strength of leadership and his guidance to this group, I think, is important for us to hear. Super pleased to have him with us today. Steve, I'll turn it over to you with all accolades. To see your involvement here is a great sign, and really appreciate your help today.
Sure, absolutely. Well, thank you, Steve, and thanks, Beth, and all of those at FNIH for your leadership for years in the biomarkers consortium space. I, of course, got involved back in my 18 years as president and CEO of the Sjogren's Foundation. Kathy Hammett, who's on this call, was instrumental and still is in leading the scientific work at Sjogren's. But, when I heard about the Gout FNIH Biomarkers Consortium idea, obviously it was important to me personally. I'm a gout patient, but being the president and CEO of the Arthritis Foundation, gout is one of our core 6 forms of arthritis that we focus on at the organization. I do want to recognize our Vice President of Research and Science, Jason Kim, who has been serving on a number of FNIH areas for us in osteoarthritis, and he's also our lead in gout. But gout is very important to us because it is not only one of the core 6 diseases that we focus on, but it is also usually our top search result on our website with so many people searching information about gout. So a lot of patients come to the Arthritis Foundation for gout information. I'll talk about that in a moment. And I thank many of the companies on this call that support our gout education and our gout awareness initiatives as well. And of course, I'd be remiss if I didn't thank ANRF and Alliance for Gout Awareness. We've worked with them over the years on various programs, and truly appreciate that collaboration, and hope that will even foster, this program will even foster more collaboration long term to help more patients and reach out to more patients. But for myself, I want to talk a little bit about my gout story, because we do have a little bit of a challenge, as you all know, in the gout space with primary care physicians versus rheumatologists. I don't want to say it's a verse, but they do treat gout differently or look at it differently sometimes. And rheumatologists don't always treat gout as always, because a lot of the patients end up at the primary care physician as first point of treatment. But for those, some on the call know my gout story, but I was diagnosed roughly in my mid-30s. I know I look like I'm still in my mid-30s, but I am not. And I ended up in the emergency room twice with gout attacks, as I call them. Horrible, horrendous pain. The first time I went, it was the classic big toe, red, air, a sheet from the hospital bed, someone touching it, no matter what it was, it would hurt even worse, including, like I said, the air or the sheet. And the ER thought I stubbed my toe and sent me home. They first put me on a morphine drip because I was in so much pain. And then they sent me home with painkillers and told me that it wasn't broken from the x-ray and it was probably fine and it would heal. And sure enough, the gout flare went down and I got better. About a year later, I had another flare, ended up in the ER. They mentioned gout, but they still thought maybe I had done the same thing running and stubbed my toe, but they thought follow up with my primary care physician. So it took about 3 years because, of course, being a stubborn male, I didn't follow up with my primary care physician right away until I had another flare. And instead of going to the ER, I went to call my primary care. It was during the day and he saw me. And he realized that, took a uric acid test and then realized I probably had gout. Being in the rheumatology space in Sjogren's, I had access to many rheumatologists. Again, being the stubborn male, I did not want to go on any medication. I wanted to kind of treat the flares as they happen. So, of course, regularly, and still to this day, I carry indomethacin and colchicine in my briefcase in case I had a flare. But then finally, thanks to Dr. Herb Bariff, who many of you might know from Maryland, and Teresa Lawrence Ford from Atlanta, both served on my board of directors, they convinced me to start to look at another treatment. So I did end up on allopurinol, which I'm still on to this day. And knock on wood, I've not had a flare since then. I am a lucky one. I've not had a flare being on that medication, but many people do. There is a compliance challenge with gout as well. And also a challenge of physicians knowing how to treat gout, both in the primary care space and then, of course, in the rheumatology space as well. And patients not understanding how serious the disease can get, how important it is to stay ahead of it and be on one of the stronger medications to make sure they prevent it from going to other joints, as well as underserved communities are also underrepresented in gout diagnosis and sometimes suffer more. And you all know about gout. Gout affects more men than women. So men are also a challenge sometimes to treat with various diseases. And so at the Arthritis Foundation, we work very hard at making sure that we have available resources 24/7, our helpline, as well as our online services for those when they're having a gout flare to find information about what it could be. But we also have webinars on gout, and also we have live connect groups that meet regularly, usually on a quarterly basis, for those that live with gout to share with each other. Mostly men join those calls. We have hundreds and hundreds of followers in our gout, even I think it's thousands of followers in that gout connect group. And it varies because usually people with gout, and you probably am preaching to the choir, usually are not worried about gout till they have a flare. And we have to change that mindset to make sure people are on drugs like allopurinol or other drugs that are out there on the market, as well as new drugs that need to be developed that work better, that are so important for this disease, and then increasing awareness so patients can get on those drugs so they don't have to have a flare, be out of work for a week, have to use crutches, not be able to move because the pain is extremely disabling. So I was, of course, thrilled being investing in the Arthritis Foundation and all of our commitment to arthritis and gout for years now, financially our commitment, but also to financially support this work. I want to thank, of course, Dan Solomon and Tony Merriman for their leadership in a new trial, how important this is that the gout space is getting attention. And I want to again thank FNIH for making sure we put in a focus on gout in this way. So looking forward to it. The Arthritis Foundation is fully committed to be supportive of this project. And you'll hear more about our financial commitment down the road. But I thank you very much, Steve and Beth, for having me speak today to share a little bit about my personal story. So I'll send it back to Beth.
Thank you so much, Steve, for sharing your story. We are excited to have you all here today. Some of you have already been involved in this project through its design phase and to where we are now. Others are hearing about this for the first time. So we're really excited to share the information with you today, and we hope you'll be excited as well. This project actually came to us. Oh, and I do want to mention, I forgot to introduce myself. I am the program manager for inflammation and immunity at the FNIH. So I am the project lead on this exciting project. And this project came to us. We do host an ideation meeting every other year in inflammation and immunity. And we had this project come through as a concept, and we're really happy with how it has progressed and to where we are right now. So I would like to introduce our project co-chairs who have been champions and supporters of this project from the beginning. We have Dr. Solomon from Harvard Medical School and the Brigham and Women's Hospital. He is a practicing rheumatologist. And then we have Dr. Merriman, who is from University of Alabama at Birmingham. He has done many years of research in gout. And then we have Dr. Hristova, who is an associate director at Amgen, and she is our industry voice and champion as well. And all three bring a wealth of knowledge and expertise, both in gout research and in the genetics and proteomics that we are implementing for this project. And we also have a project development team that has been working with us over many months, which is inclusive of biopharma companies, as well as biotech, platform therapeutics, academia, patient advocacy organizations, and of course, our partners at the NIH. And now I will turn it over to Dr. Solomon, will tell us a little bit of the background of this project and this disease and the clinical trial that we are leveraging. And then Dr. Merriman will continue with the project aims. So, Dan?
Great. Thanks, Beth, and thank you very much to the Steves for that introduction. I think that many on the call know a bit about gout, but just to review some of the key areas, it really is the most common inflammatory arthritis that we see in rheumatology and that exists. Gout occurs in people with elevated serum urate levels, hyperuricemia, who have crystals deposited in and around their joints. And the flare, as Steve mentioned, which is incredibly painful, flares occur when the innate immune system reacts to crystals, and there's a inflammasome activation producing huge amounts of IL-1 beta. And unfortunately, they're unpredictable and described as tremendously painful, only worse being childbearing. And they're unfortunately still a bit poorly understood by patients and most clinicians. And so what Steve described before is kind of his journey to get a diagnosis and to get on the right treatment is a really, really common story. I was in clinic this morning and unfortunately saw somebody who spent a long time bouncing around emergency departments before he got sent to a rheumatologist and got a diagnosis, and now is under careful care and only needs to see me every once in a while when he needs a refill on his allopurinol. So why don't we go on to the next slide? This is kind of some representative pictures. On the left is the ankle that we often see in somebody who presents to the emergency department. with an acute flare. In the center is the person who has very end stage tophaceous gout, which affects the digits in the hands or the feet, and there's the kind of the classic needle-shaped birefringent crystal that we see on polarized microscopy. Next slide. So we know that it's the most common inflammatory arthritis, affecting about 5%, and it's growing in prevalence. About 12 million adults in the US are affected. It has high rates amongst Asian American and Black American, and the hospitalizations for gout actually exceed those for rheumatoid arthritis, which is important, and is the chart on the right that we're showing. And as was mentioned, the optimal care is debated, and this is the reason that we went about pursuing a trial, which I'll explain on the next slides, called TRUST. But the optimal care is debated between rheumatologists and internists, between treat to target serum urate versus what the American College of Physicians has advocated, which is treat to avoid symptoms. And as you can imagine, rheumatologists believe that many more people with gout need to be using chronic urate-lowering therapy with agents such as allopurinol or febuxostat or pegloticase or other agents that are in development. And it's true that there really has been very little pharma development in this area. But there are some new agents that are being worked on currently and promise to really help patients. And as I said before, gout impacts the inflammasome, and it activates IL-1, IL-6 pathways, which are interestingly important for many other conditions, including autoimmune diseases, cancer, cardiovascular disease, renal disease, and diabetes. And so this pathway that gout is kind of known to impact is really a pathway that is active in many other conditions. And we think that this is one of the reasons why this biomarker effort will be so important and fruitful, not only for gout, but for a variety of conditions. Next slide. Again, there's a huge unmet need. Clinical need includes understanding which patients are more likely to flare. So many patients with hyperuricemia never have a gout attack. And then some patients with gout will only flare once a year, but many patients flare 3 or more times. I mean, I've encountered patients who have kind of constant flares, the chronic polyarthritis of gout. We need to figure out who to offer more intensive management, which patients, and as you can imagine, patients who flare frequently should be the targets of such treatments. And reducing flare frequency will improve outcomes for not only the patient, but the whole family who has to deal with these attacks and the loss of work and disability. Urate-lowering therapy is effective for over 90% of patients, but we still know that 1/4 to 1/3 of patients have severe flares with functional disability, TOFI, or emergency department visits. 10% of patients have over 3+ flares per year. And while we can often achieve a target urate of 6 milligrams per deciliter or below, flares still happen. And in a recent trial that was run through the VA, 40% of patients who met the target urate level still had a flare. There's emerging evidence that flares increase the short-term risk of cardiovascular events. So patient management of these cardiometabolic comorbidities is difficult, but they're common amongst patients with gout. Next slide. So the TRUST trial is what we've been alluding to. This is an NIH-funded trial funded by NIAMS that I'm one of the co-PIs of with Hyun Choi and Michael Berry, 2 clinicians at Mass General. And it's a 24-month trial. It's a simple 2-arm strategy trial comparing treating to a target serum urate of less than 6 versus treating to avoid symptoms with anti-inflammatories and sparing use of urate-lowering therapies. The primary aim is to look at the gout flare frequency over the 2 years with quality of life, being a secondary outcome. And then we have established sub-studies on glycemic control, blood pressure control, renal function, and working on a coronary CT angiography sub-study contract at the current time. So a variety of interesting aims, and we're now into the trial where we were able to get $9 million through the NIH and $2 million from another pharma company. And so what we're trying to do with this this FNIH project is really leverage the trial structure of TRUST and the ability, the infrastructure to collect biospecimens. We already have enrolled about 45 patients, and we're just bringing on 4 more sites in the next month. So that is going to start to accelerate now. So that's the status of TRUST that's so important for the backbone of this current biomarkers project. Next slide. So again, the TRUST trial objectives, the primary aim is to look at gout flares and quality of life, comparing these two strategies. And the secondary non-articular sub-studies, as I mentioned, blood pressure control, glycemic control, kidney function, and cardiovascular disease. And the clinical data that's being collected as part of the trial include the demographics, comorbidities, co-medications, body mass index, blood pressure, kidney function, and gout history. Next slide. So I'm going to turn it over to Tony to talk a little bit more about the FNIH project aims and objectives. Tony, go ahead.
Thanks, Dan. Before I start on this, I thought I might give you a little bit more of an intro about myself. I'm, as you might have figured out already, not from the South. And in fact, I'm not even from the US, but I grew up in New Zealand and recently shifted to UAB to carry on my gout research program. But in New Zealand, gout is a very, very big issue in some communities. The prevalence is in Maori, for example, 15%. And I got about 20 years ago now, got convinced by my colleague, Dr. Nicola Dalbeth, who's a world-leading, what we call a goutologist, to transfer my genetic studies from rheumatoid arthritis to gout. And I have spent many days in those communities talking to patients. And Steve Taylor, I really appreciated what you had to say, and I've heard many stories like that. And the strategies needed for convincing men, and men in their 30s to get on to allopurinol, we're still working them out in New Zealand, so it is a challenge. Anyway, coming back to this project, the aims and objectives. So the first prerequisite we need for this to establish a specimen biobank from which we would draw the specimens and generate the genomic and genetic data for the primary objective, which is to develop a predictive model for people who have frequent flares. And I certainly like to thank the Arthritis Foundation for stepping in and funding and helping us to establish this specimen biobank. And we're right in the middle of working all of that out. So there are 2 aims. So what we want to do is look at samples from people from the biobank and look at baseline, then collect their data. We also want to look at 12 months and then collect data on flare frequency. And we want to compare people who flare frequently, which we have defined as 3 or more or greater than 2 a year, to people who flare infrequently. And in aim 1, we will develop the markers that we have settled upon as part of developing the concept document with the working group in FNIH. We will have genetics in there. We will have gene expression, looking at total RNA and microRNA, and also the proteome as biomarkers. And what we will do in aim 1 is test and validate in separate trust sample sets to identify biomarkers that distinguish people who frequently flare as opposed to those people who don't. And then we will test out this group of markers as being predictive for frequent gout flares. We'll use samples in the trust trial as our internal cohort, but we plan to validate externally in the stop gout trial, which Dan mentioned in his introduction. And one advantage about the stop gout trial is that we already have data, gene expression data from that trial, and that's an auxiliary RO1 led by Dr. Richard Reynolds, who's on this call and is my scientific partner down here at UAB. The next slide, please, Beth. So the analysis plan, we will identify two groups of 50 patients each from the trust trial, and each group will be comprised of 25 patients who have no or low flares versus 25 patients who we define with frequent flares. We will compare samples at baseline and 12 months. What we will do is pull those samples, generate the transcriptomic data through bulk RNA sequencing and microRNA, and thanks to the efforts of Beth and FNIH, we have some really good pricing for that to carry on with that. We will generate proteomics data using the Olink 3072 Explore panel, which has 3,072 potential proteomic markers in there. And co-chair Vinci is our expert in proteomics, and down here at UAB, we are expert in genetics and transcriptomics. And as I described in AIMS, we will identify in AIM1 the differentially expressed markers in the discovery of a group and validate in the other group of 50. And then in the predictive model, we will combine all of the 100 patients from the trust trial and develop a predictive model in that. And then we will validate it in the stop gout trial using some data we already have, and we would also need to generate some proteomic data. And I think it's just to emphasize how important it is for these auxiliary studies and clinical trials for us as geneticists, the great quality of the data that people get from these trials. It means particularly we can be very confident about our outcome data in these trials. Next slide. Here is a cartoon of RNA sequencing. So in our study design and in gout, it's very convenient that we can use that whole blood would be our tissue we would look at. We would do two separate analyses, generating total RNA and also microRNA separately, which is also an important in regulating the immune system. Next slide. And here's the Olink proteomics workflow, which the PEA workflow, which is stands for proximity extension assay. So basically, you have in your antibody-based immunoassay, there are two antibodies that simultaneously bind to a target in solution. They bring oligos together, which are amplified, and then a library is prepped by PCR. And next generation or high throughput sequencing, or these days it's standard sequencing, we can detect the relative levels of proteins in a particular plasma sample. And as I say, Vinci is the expert on this, and we're very grateful to have her as a co-chair. In the context of use, so these are prognostic biomarkers, and in terms of applying this and making patients' lives better, what we would envisage is being able to screen patients relatively after they have gout. And if patients can be predicted to be more likely to flare frequency, then they can be offered more intensive management, more intensive education as well, for which is a crucial part of convincing this demographic of men to take a drug like allopurinol for the rest of their lives. And Also, importantly, it should allow more efficient drug trial designs to focus on high risk patients for developing better drugs for treating flares. And currently, the drugs for treating flares are very non-specific, and being used more frequently, quite frequently, can be toxic in the long term. And so there is a focus for better drugs in for treating flares, and if we can identify a high risk group, that will make the clinical trials more less expensive and better powered. And I would just like to also point out that in this particular study, we will be actually generating new knowledge on potentially new drug targets in their own right as well, by potentially identifying newly identified biomarkers that maybe we could validate as drug targets. And that's, I think, Beth, was I going to run through the slide? I think I was, wasn't I?
Yeah.
Yep. And I probably said a bunch of this stuff already. So the direct impact, earlier intervention, improved patient stratification. The biobank itself, you should emphasize that Arthritis Foundation funding will be available for the gout community to use for future gout research. And one of the most important types of sample we are collecting in that bank is we're cryopreserving PBMCs, which would be available for future single cell RNA and single cell studies later on, including proteomic potentially, and also epigenomic studies for future studies. And Dan already mentioned the benefit, and I can emphasize here, and in the bottom right, this is what we are spending a lot of time studying down here at UAB, the factors that are controlling, and on the big purple blob, in here that I'm circling, I don't know if you can see that, but the inflammasome is a really important piece of molecular machinery that's activated in the innate immune system, and that's what the combination of phagocytosis of MSU crystals down the bottom and activation of the TLR receptors up the top here activates this inflammasome, and it's a huge area of research at the moment, understanding the factors involved in activating and regulating the activity of this inflammasome and turning it off. But gout itself is the best acute model, a clinical model of inflammasome activation that there is. And, we will be able to generate new knowledge on the pathways that happen there, which will be also applicable to understanding how, for example, a gout flare, how does it actually increase CV, cardiovascular disease events, or heart disease events in the 3 months following a flare? There's data coming out on that. So potential new targets, I've mentioned that, but destigmatization really is a underappreciated impact of biological research in gout, that being able to talk to patients and explain what happens and explain that there's a considerable amount of inherited innate biology here on causing these gout attacks, hyperuricemia in response to crystals. And certainly from my experience in New Zealand, there's a lot of stigmatization, and to help patients understand that, hey, it's not your fault, guys. And then that's a good basis for primary care physicians to talk about allopurinol. And then finally, potential submission to the FDA biomarker qualification program.
Thank you, Tony. I will wrap it up with just an overview of the project budget and timeline that we are looking at. So we are fortunate, as mentioned, to already have the biobank funded by generous support from the Arthritis Foundation. The rest of the project, for all of the technologies, for the genetics and proteomics, would be about 1.6 million. We are looking to have a minimum of 3 partners, hopefully, on this project. Obviously, the more partners that we have joining us financially will bring the cost down for all. And also, we are hopeful that with our patient advocacy partners as well. There will be a benefit here as part of this project to, really make sure that we benefit the patient community, get the word out. This is a newer area for, our industry partners. So we're hopeful that this will just kickstart that in the right direction. If we are hopeful to over fundraise and have more partners interested, we do have our project development team has identified a couple areas of additional research if the funds are there. We would like to look at a personalized approach to the urate lowering therapy using our prediction model to look at response to allopurinol and which dose would be required to achieve that serum urate level target of 6 or less than 6. And then we also could have an aim to look at predictive markers for treatment response for those that have a high propensity for flaring that we identify within those first 12 months to look at the treatment over time over the next 13 to 18 months as a predictive biomarker. Those are also additional research opportunities. And currently, we are in project development. We are in our final stages of the project plan approval, and we'll be starting our fundraising process. We are hopeful to launch this project by the end of the year, fully funded. And then it will be a 3-year project starting. Obviously, we will be collecting samples that are coming from the TRUST trial for the biobanking throughout the entire length of the project. And then once we have enough samples accumulated, we will start aims 1 and 2 for processing and then analysis. And there are many benefits to partnering with FNIH. The first being early access to the data. You would have early insights to this, multi-omics level data, including all of the cardiometabolic and renal function data that we're collecting as part of the TRUST trial. Using those clinical insights for patient identification and stratification for clinical trials. Also potential therapeutic targets related to the inflammasome for gout and other related diseases as well that involve the inflammasome. This is a team science approach, so it is cost sharing among multiple organizations, but you still get the value of the access to the data before it is made public. It's an opportunity to be part of new research in this field and be part of that contribution and in the forefront of that. And also, patient-centric. We are focusing on destigmatizing gout, making the awareness that this is biologically based, and really reducing that stigma for the patient community for those that are living with gout. And also the opportunity to engage and partner with experts across government, academia, other industry members, nonprofits, and patient advocacy. So the next steps, we do have one final approval that we need to secure from our biomarkers executive committee, and that will be next month in September. The FNIH does rely on support from the private sector to fund these projects. So after this webinar, we will be calling on all of you to provide your insight and any tweaks or modifications you think we should be making to the project plan. That is a very important part of this approval process as well. Just letting us know your feedback, your thoughts on the project, and how willing your organization would be to support as well. And I believe now we will open it up for questions. Thank you all so much.
If I wanted to jump on and maybe perhaps help stir some of this, and this maybe go
Please.
back to Dan and Tony directly, always an important component in our approval process. And I do think as we go up, not only through our steering committee, but our executive committee, is, we often have what looks to be really great science exactly meeting the unmet need and where the focus is. But what is, I know you presented, buy up other things that we can do very much for champion these things internally, the organization, those might be important pieces of this. And if that is something that would strengthen the project, make it more palatable or able to champion with an organization, if it did Y or Z other than the X that we're proposing, I think that's important too. So I asked kind of Dan and Tony, in addition to what you have here and what you might propose, what are the additional challenges? What are the additional maybe gaps in the research in the gout field that, whether we have the dollar or not, that are still out there that maybe this doesn't address? And maybe that stirs a couple questions from other groups that says, here's where our pipeline's focused. This doesn't address this. Maybe we could.
Yeah, I'll start and then Tony jump in. But I think the some of the secondary aims that we have. So we have 650 people being enrolled overall to the TRUST trial for our primary aims, and we'll enroll several hundred people into all the sub-studies on glycemic control, on kidney function, on blood pressure, and on coronary CT angio. And again, we know those are really important in frequent cardiometabolic complications of gout and likely have overlap with the same pathobiology, with the inflammasome and other downstream cytokines. And having the several hundred people being involved in each of those sub-studies gives us the ability to not just look at the intermediate endpoints, HbA1c or blood pressure, but also some actual endpoints, new onset diabetes, new onset cardiovascular events, et cetera. And we're going to have the biospecimens in the can in a prospective manner. So I think that this is really going to be exciting. Science is going to be generated from these samples with all the paired phenotypic information on these patients who unfortunately have a lot of comorbidities. It's going to be an important group of patients to study for gout as well as these other inflammasome related conditions. I don't know, Tony, if you want to add anything to that.
No, I mean, add about the third aim that we mentioned, allopurinol dosing, and it's quite clear now, and I've been involved in studies here, that there is at least a genetic contribution to the ultimate dose of allopurinol that a patient will need to get to their target of 6. And the treat to target arm of TRUST will provide us good information on, which patients get treated to target with a standard dose, which is 300 milligrams, and which patients will require a larger dose than that. And part of what we will be able to do is potential in aim 3 is look at biomarkers, genetics and proteomics and RNA expression that might be able to predict patients who will require a larger dose of allopurinol, and perhaps that in the clinic and making patients' lives better means that in the future that those patients could be got to target quicker for their gout. So that's all I would have to add to what Dan said.
My name is Gary. I do want to jump in and just say thank you to the panel. Just listening to you guys and what's going on is really remarkable. I'm excited. Just a little bit about myself. I'm Gary Ho. I am a gout patient. I actually experienced my first attack at the age of 24. And then spent the next 16 years trying to get a diagnosis because for whatever reason, it seemed like I fell outside of what they thought, a person who had gout would look like. So that was in 1994. It wasn't until 2010 when I actually received that diagnosis, which was chronic gout. I was able to participate in a clinical trial at the time with my rheumatologist, an infusion medication at the time. That brought me to a point where I was able to take allopurinol and then ultimately Uloric, and I brought down my uric acid level to a point where it's manageable. And like Steve and Steve Taylor, thank you for your story. I really identify a lot with what you said. Since 2010 and being able to manage my uric acid level, I've been pain-free. I started a group, the Gout Support Group of America, with my rheumatologist. We're about 16,000 members or so on Facebook. I'm really excited to go back and talk to our group about how we might be able to support. I don't know, right? But I know that what you're talking about just hits a core. I mean, there's so much lack of information on gout. The fact that when I jumped into this arena, there was about 8 million gout patients. Now we're talking about 12 million. It's not going in the right direction, obviously. When we talk about people living with gout, oftentimes people downplay the people that have gout, downplay the severity of their sickness, right? They don't understand that they're sick. I often hear that, hey, I only have 2 or 3 attacks per year. I'm okay. What they fail to realize is 2 or 3, you're in the chronic stage, right? I mean, that's pretty bad. So there's a lot of need for education, especially also with, I think, personally, with the general practitioners out there that play gatekeeper, right, to the patient community, right? And so we need to be able to educate them, possibly through what you're talking about, new biomarkers, right? If you're able to figure something new out and you're able to pass the nuggets of truth over to this new generation of physicians out there to treat patients, that's going to be a win-win situation. So, I mean, I'm really excited to see what's happening. I'm not sure how the Gout Support Group of America can support, but we have access, again, to thousands of gout patients in America, in the English-speaking world. And, I'm big on collaboration, so I will be happy to, go back to our team and talk. But I do want to say thank you for what you're doing.
Gary, I'll say that I think we've been in touch, and I think the Trust Trial has reached out to your organization and are hoping to collaborate on trying to recruit some of the people that are in your network to be part of the trial. And so I think you actually are already supporting us and didn't realize it.
Okay, wonderful. Wonderful. Thank you.
Thank you.
Thank
Yeah.
you. Thank you for sharing. I also wanted to let our other project chair, Vency, if you had anything else to add or share about, the science of it, the technologies that we're using. Just wanted to give you a chance to speak.
So I want to say also thank you for looping me, and I was brought into the team earlier this year. So I'm fairly new, but I'm glad for this opportunity to work with not just a team of gut experts, but also patient advocacy groups and patients. It really makes a difference to what I do. In terms of the technology, I would say, in my opinion, from what I have seen in my past as well, the integration of multi-omics is key for a lot of the innovative and better biomarker strategies. I think it's a great opportunity. So happy to be part of it. I think this is a wonderful study and a great effort, and hopefully will address the huge unmet need in gout.
Thank you. Any other questions for our co-chairs while we have them? Well, if not, I want to thank you all so much for joining, and we will definitely be following up. Myself and other colleagues at FNIH will be in touch to get your feedback on this, to see where we can maybe align with your organization to support this project to fruition.
Awesome. Thank you.
Thank you all.
Yes.
Thanks, Beth. Thanks, Steve. Thanks, Tony and Daniel.